Okay, we'll be looking at the BioSci 140 Lab 19, the glucose tolerance test laboratory. We'll first look at how the data is collected. So for the laboratory, we'll take initial blood sugar reading. The participants will not necessarily be fasting. They will then consume 80 grams of sugar. There will be Jelly Belly jelly beans. Each Jelly Belly jelly bean is approximately 4 calories, which means since it only contains sugar and flavoring, they will consume approximately 80 Jelly Belly jelly beans. And then once we take that initial blood sugar reading, we'll continue to take blood glucose approximately every 30 minutes until they have recovered and their blood glucose is back to normal. Usually that's within 3 hours. We'll call the fasting glucose T0. T is for time. The glucose tolerance test will be measured on just one of the what they call glucometers. It measures glucose. The machines just require a small amount of blood and then automatically measure glucose. It's the same type of machine that somebody with diabetes would potentially use. Once we have started the test, we'll expect the subject to peak somewhere in 30 to 60 minutes in terms of their blood glucose, and then come back down, and usually within 3 hours it's returned back to below 100, which would be a normal test. Here's an example of what we can see with a normal test. Note the subject starts out below, right here, below 100. Normal blood glucose is actually between 70 and 99 milligrams per deciliter. Note they reach a peak fairly soon, in this case within about a half hour, and they're back below where they started within 2 hours. Someone who did not respond properly, note here their blood glucose is close to 125, probably a little above, as diabetes is measured as a fasting glucose of 126 milligrams per deciliter or above. Note that it takes them a while to peak, and they peak somewhere in about an hour and 30 minutes. Even after 2 hours, they're still at a fairly high blood glucose, way over 200, and if you extended that out to 3 hours, they'd still be fairly high. So that would be a abnormal test that we're looking at there. Here's some examples of the type of figures we'd expect. In class, we'd expect to see a normal fasting blood glucose right here, below 100. It would peak within 30 or 60 minutes, so notice the peak is within about a half hour. The peak's only at about 125 or so, and then within an hour and a half it's back below 100, fairly normal. For someone who is pre-diabetic, prediabetes is identified when somebody has a fasting glucose between 100 and 126 milligrams per deciliter. Note here that they start above 100, right, and then peak after about an hour, hour and a half, and still after 2.5 hours on this graph, they're still way above 100. The third graph, the triangular graph, is someone who is diabetic. They start with their blood glucose excessively high. In this case, it's 150 something. They still haven't peaked after an hour and 50 minutes, and their blood glucose is excessively high. So we'll see somebody like this with diabetes. This is the type of response. We would not expect this response in class because we would ask anybody who was a known diabetic to not participate. These types of graphs we could obtain if someone had some liver disease or something, as that helps control blood glucose. but the vast majority of cases, when we see something like this, it would be something with diabetes, which would be a problem, either making insulin or responding to insulin. So that completes the portion of the laboratory where we just kind of collect data, and we'll move on to discussion next.